Now the next part in DNA, the nucleic acids, is the replication of DNA. What is replication? Production of exact copy is called actually replication. Replica, the Xerox copy of the original. That is called replication. Now it is one of the important properties of nucleic acid. It is happening during the life of an organism, then only during cell division. The DNA amount is maintained during mitosis or in the case of meiosis also. Now, during replication, the parent DNA produces exact copy of its own. That is called Xerox copy, exact copy of its own. You know that one, in the case of DNA molecule, we have complementary base pairings in the two strands of DNA. So, the nucleotides of one strand act as template for the formation of the nucleotides of another strand. After cell division, the genetic information contained in the daughter cells are exactly similar to those of the parental cell, or the nature of the DNA molecule in the case of parental cell and the daughter cells would be similar. So different methods are adapted, different mechanisms are explained by different people. But the most important mechanism explained by Watson and Crick was semi-conservative method in 1953. That means other methods are conservative method, dispersive method. Now this is semi-conservative method. What is semi-conservative method of replication? Now here I am taking the parental DNA, strand A, strand B with the same colors. So each strand is acting as a template or model. So during replication. So the parental strand now A synthesizes its complementary strand B. Similarly, the parental strand B synthesizes its complementary strand A. So as a result, one DNA molecule gets converted into two DNA molecules. These two DNA molecules are exactly similar to those of the parental strands. But here, if you are taking this DNA molecule, out of the two strands, one strand is the parental strand which is conserved, no change. Another strand is a newly formed strand. Likewise, here you see A produces B, B produces A. So out of the two strands, one strand is conserved and another strand is the new one. So this is called semi-conservative method. One strand, one of the parent strand is conserved, the another strand is a newly formed. So in a daughter molecule, one strand is a old strand or the parent strand, another strand is a new strand, hence called semi-conservative method of replication. Now what are the different steps taking place during semi-conservative method of replication as proposed by Watson and Crick in 1953? The first step is the initiation process. What is happening? There is a site where the replication starts. That site is called site of origin of replication. Site of origin of replication. So we need origin of replication site. At that site, the two strands open and separate to form a fork-like structure, what is called replication fork. At the site of origin of replication, now the two strands get separated and open up so as to form a fork-like structure, a bifurcated structure called replication fork. The two strands are normally separated by the action of enzyme number 1 helicase. It breaks the hydrogen bonds so that the two strands being separated. Now there is another enzyme what's called topoisomerase. Now there are certain regions above the replication fork. they are normally in the form of twist in the unwinding process. Or we can say supercoil. It is lying above the replication fork, they are in the form of twist, they are not being separated. And these strands are separated by means of topoisomerase. It relieves the tension or actually the twist in the supercoil. That is topoisomerase. Now, once replication fork is formed, now the process of replication begins. But normally, the process of replication is brought about by DNA polymerase. That is elongation of the strand. But the DNA polymerase cannot initiate the replication process. It needs the help of somebody. To begin with, a small segment of RNA, that is a few nucleotides of RNA, are laid down at the origin of replication, and that is called RNA primer. This is called RNA primer. This RNA primer is produced by an enzyme, RNA polymerase, but in the book it is given, it is produced by the DNA template. The out of the two strands, each strand is acting as a template for the formation of new strand, the mod. Now, the RNA primer is laid down, a few nucleotides of RNA, by the enzyme RNA primase, or simply primase enzyme, the third enzyme, for the formation of, that is, primer, which initiates the elongation process. Now, the enzyme DNA polymerase operates, adding the nucleotides one after another to the RNA primer, so that the chain elongation occurs. That is brought about by the DNA polymerase. Now, out of the two strands, one strand is formed continuously, that is from the terminal end towards the replication fork, this one, from the terminal end towards the replication fork. The strand which is formed continuously towards the replication fork is called as a leading strand. This is a leading strand. The leading strand is synthesized by DNA polymerase of one type. In another strand, the synthesis of DNA is not continuous. The strand is not synthesized continuously. It is being normally synthesized in the form of fragments. And that strand where the synthesis of DNA is discontinuous is called the lagging strand. Lagging strand. Now in the lagging strand we have the fragments of DNA. Such fragments of DNA are called Okazaki fragments. Okazaki fragments. Now the synthesis of lagging strand occurs away from the replication fork. That is from the replication fork to us. That's why just I showed in the form of broken fragments. These fragments are called Okazaki fragments. And the synthesis occurs away from the replication fork. And these fragments are joined together to form a continuous strand with the help of another enzyme, what is called DNA ligase. Ligation means stitching, joining or pasting. The DNA ligase joins all the fragments which are formed discontinuously, namely the Okazaki fragments, to form a continuous strand. So, one strand is synthesized continuously. That strand is called as a leading strand. Another strand is actually synthesized discontinuously. That is called a lagging strand. The synthesis of lagging strand occurs away from the replication fork, and the synthesis of leading strand occurs towards the replication fork. Now the process of replication stops when what is called the replication fork of the two strands meet in a region. That region is called the terminus region. This is the terminus region. The replication fork of the two strands meet at a region, what is called terminus region. This terminus region is present opposite to the origin of replication. Now this is the place of origin of replication. And now this is what is called the place of terminus, the stopping of the replication process. So anyway, in the semi-conservative method, out of the two strands produced, now we have just one DNA molecule. This is another DNA molecule. One strand is a parallel strand. Another one is a newly formed, what I represented in the previous picture. So two DNA molecules are exactly similar to those of the parents are produced, and they have the same genetic information as that of the parent cell. Now these are all the 5 enzymes involved in replication process. So this method of semi-conservation was proposed by Watson and Crick in 1953. They were awarded Nobel Prize for this one in 1962. So that's all about the DNA structure and replication of DNA. Thank you. Hi students, regarding the replication of DNA, I have drawn a symbol diagram on the board as per the book, how to draw the diagram. Now you see that one, it is being formed of 2 strands. This is one strand, this is another strand. One strand is running in 5' 3' direction, another strand is running in 3' 5' direction. Now this is the origin of actually replication, the site of origin of replication. Now the 2 strands being separated by what is called the helicase enzyme, so as to form a replication fork. Now this is the replication fork. This is the replication fork. So with the help of helicase enzyme, the 2 strands get separated. Another enzyme, topoisomerase, what I mentioned, responsible for just actually remove the twist lying above the replication fork during the replication process. Now, one of the strands, normally this is a template strand, parallel strand, 5' 3', another template strand, 3' 5'. In one of the strands, now it acts as a template where the DNA is produced actually discontinuously. The strand which is formed discontinuously as broken segments is called lagging strand. The fragments are called Okazaki fragments. At a later stage, these fragments are joined together by means of DNA ligase to form a continuous strand. So, to begin with, the strand is formed discontinuously as broken fragments. These fragments are called Okazaki fragments. Later, they are joined by the enzyme DNA ligase to form a continuous strand. Now, this strand is produced away from the replication fork. Now, this is the direction of the formation of the lagging strand. Another strand is formed continuously, which is normally formed towards the replication fork, and this is called the leading strand. Now, the addition of nucleotides, that is called polymerization, is brought about by the enzyme DNA polymerase. DNA polymerase, this is a DNA polymerase in both the strands. But the DNA polymerase cannot initiate the process of replication. So, a few nucleotides of RNA is added at the point of just replication of what is called the lagging strand, or we have what is called the leading strand, and this is called primer. A few nucleotides of RNA are being laid down as a small fragment called primer, and that is followed by the addition of nucleotides in both the strands. Now, this primer is produced by what is called DNA primase of the DNA template. So, this is a diagram showing how far you have to draw the diagram in a simple way, because the diagram is given in a colorful way, a complicated way given in the book. So, 2 parallel strands, lagging strand, leading strand, Okazaki fragments. Now we have the DNA polymerase, the fragments are joined together by DNA ligase. Now we have the formation of primer, a few nucleotides of RNA, and that is being produced by the enzyme DNA primase. The helicase is responsible for separating the 2 strands by breaking the hydrogen bonds, and the topoisomerase is responsible for just making the untwisting of the segments lying above the replication fork. So, this is a semi-conservative method of replication. I am drawing this diagram for your reference to draw it in a simple way. What is the significance of DNA? So DNA is the storage of hereditary information, responsible for the transmission of hereditary information from one generation to another, as it is contained in the genes. Number 2, the blueprint, the information, the code is present in DNA for the synthesis of protein, and that genetic information that contained in the DNA is responsible for the expression of protein molecule by means of transcription and translation processes. We'll be studying in higher classes. Then it also controls the developmental processes, the embryological development, and also the various life activities. The various life activities and the developmental process of an organism are also under the influence of DNA molecule. Thank you.